So here we are starting with a new chapter, chapter 3, human reproduction. As you are aware, humans are sexually reproducing and viviparous. The reproductive events in humans include formation of gametogenesis, sperm in males and ovum in females, transform of sperms into the female genital tract, insemination, and fusion of male and female gamete fertilization leading to formation of zygote, thus is followed by formation and development of blastocyst and its attachment to the uterine wall, implantation, embryonic development, gestation, and delivery of the baby, parturition. You have learned that these reproductive events occur after puberty. There are remarkable differences between the reproductive events in the male and in the female. For example, sperm formation continues even in old men, but formation of ovum ceases in women around the age of 50 years. Let us examine the male and the female reproductive systems in human. The male reproductive system. The male reproductive system is located in the pelvis region. It includes a pair of testes along with accessory ducts, glands, and the external genitalia. The testes are situated outside the abdominal cavity within a pouch called scrotum. The scrotum helps in maintaining the low temperature of the testes, lower than the normal internal body temperature necessary for spermatogenesis in adults. Each testis is oval in shape with a length of about 4 to 5 cm and a width of about 2 to 3 cm. The testis is covered by a dense covering. Each testis has about 250 compartments called testicular lobules. Each lobule contains 1 to 3 highly coiled seminiferous tubules in which sperms are produced. Each seminiferous tubule is lined on its inside by two types of cells called male germ cell, that is spermatogonia, and Sertoli cell. The male germ cells undergo meiotic divisions, finally leading to sperm formation, while Sertoli cells provide nutrition to the germ cells. The region outside the seminiferous tubules, called interstitial spaces, contain small blood vessels and interstitial cells or Leydig cells. Leydig cells synthesize and secrete testicular hormones called androgen. Other immunological competent cells are also present. The male sex accessory ducts include retor testis, vasca efferentia, epididymis, vas deferens. The seminiferous tubules of the testis open into the vasa efferentia through retor testis. The vasa efferentia leave the testis and open into epididymis located along the posterior surface of each testis. The epididymis leads to vas deferens that ascends to the abdomen and loops over the urinary bladder. It receives a duct from vesicles, seminal vesicles, and opens into urethra as the ejaculatory duct. These ducts stores and transport the sperms from the testis to the outside through urethra. The urethra originates from the urinary bladder, extends through the penis to its external opening called urethral meatus. The penis is the male external genitalia. It is made of a special tissue that helps in erection of the penis to facilitate insemination. The enlarged end of penis, called the glans penis, is covered by a loose fold of skin called foreskin. The male accessory glands include paired seminal vesicles, uptwisted and paired bulbourethral glands. Secretion of the glands constitute the seminal plasma, which is rich in fructose, calcium, and certain enzymes. The secretion of bulbourethral glands also help in the lubrication of penis. The female reproductive system. The female reproductive system consists of pair of ovaries along with a pair of oviduct, uterus, cervix, and vagina, and the external genitalia located in the pelvic region. These parts of the system, along with a pair of mammary glands, and integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth, and childcare. Ovaries are the primary female sex organs that produce the female gamete ovum and several steroid hormones, ovarian hormones. The ovaries are located one on each side of the lower abdomen. Each ovary is about 2 to 4 cm in length and is connected to the pelvic wall and the uterus by ligaments. Each ovary is covered by a thin epithelium which encloses the ovarian stroma. The stroma is divided into two zones, a peripheral cortex and an inner medulla. The oviduct, fallopian tubes, uterus, and vagina constitute the female accessory ducts. Each fallopian tube or the oviduct is about 10 to 12 centimeter long and extends from the periphery of each ovary to the uterus. The part closer to the ovary is the funnel-shaped infundibulum. The edges of the infundibulum possesses finger-like projection called fimbriae, which help in collection of the ovum after ovulation. Infundibulum leads to a wider secretion of some factors which help in the processes of spermatogenesis. Let us examine the structure of a sperm. It is a microscopic structure composed of a head, neck, middle piece, and a tail. The plasma membrane or up plasma membrane envelopes the whole body by the sperm. Of sperm, the sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by a cap-like structure acrosome. The acrosome is filled with enzymes that help in fertilization of the ovum. The middle piece possesses numerous mitochondria which produce energy for the movement of tail that facilitates sperm motility essential for fertilization. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for a normal fertility, at least for 60% sperm must have normal shape and size, and at least 40% of them must show vigorous motility. Sperms released from the seminiferous tubules are transported by the accessory ducts, secretion of epididymis, vas deferens, seminal vesicles, and a plasma membrane. And a plasma along with the sperms constitute the semen. So sorry for the one-line missing. Seminal vesicles and prostate are essential for maturation and motility of sperms. The seminal plasma along with the sperms constitute the semen. The function of the male sex accessory ducts and the glands are maintained by the testicular hormones androgen. The process of formation of a mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gametes, mother cells, oogonia, are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into prophase 1 of the meiotic division and get temporarily arrested at that stage called primary oocytes. Each primary oocyte then gets surrounded by a layer of granulosa cells and is called the primary follicle. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles and left in each ovary. The primary follicles get surrounded by more layers of granulosa cells and a new theca and are called secondary follicles. The secondary follicle soon transforms into a tertiary follicle which characterized by fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and outer theca externa. It is important to draw your attention that it is at this stage that the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. It is an unequal division resulting in the formation of a large haploid secondary oocytes and a tiny first polar body. The secondary oocyte retains bulk of the nutrient-rich cytoplasm of the primary oocyte. Can you think of any advantage for this? Does the first polar body born out of the first meiotic division divide further or degenerate? At present, we are not very certain about this. The tertiary follicle further changes into the mature follicle or Graafian follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The Graafian follicle now ruptures to release the secondary oocyte or the ovum from the ovary by the process called ovulation. Can you identify major differences between spermatogenesis and oogenesis? Spermatogenesis एंड oogenesis के बीच में differences. Schematic representation is given. क्रोमोसोम नंबर पर सेल दिया हुआ है एंड स्पर्मेटोगोनिया और oogonia दिया हुआ है. Now let's come to the menstrual cycle. The reproductive cycle in the female primates, example monkeys, apes, and human beings, is called menstrual cycle. The first menstruation begins at puberty and is called the menarche. In human females, the menstruation is repeated at an average interval of about 28 to 29 days. And the cycle of events starting from one menstruation till the next one is called the menstrual cycle. One ovum is released, ovulation during the middle of each menstrual cycle. The major events of the menstrual cycle are shown in the figure 3.9. The cycle starts with the menstrual phase. When menstrual flow occurs and it lasts for 3 to 5 days, the menstrual flow results due to breakdown of endometrial lining of the uterus and its blood vessel which forms liquid that comes out through vagina. Menstruation only occurs if the released ovum is not fertilized. Lack of menstruation may be indicative of pregnancy, however, it may also be caused due to some other underlying causes like stress, poor health, etc. The menstrual phase is followed by the follicular phase. During this phase, the primary follicles in the ovary grow to become fully mature. The Graafian follicle and simultaneously endometrium of uterus regenerate through proliferation. These changes in the ovary and the uterus are induced by changes in the levels of pituitary and ovarian hormone. The secretion of gonadotropins LH and FSH increases gradually during the follicular phase and stimulates follicular development as well as secretion of estrogens by the growing follicles. Both LH and FSH attain a peak level in the middle of cycle about 14th day. Rapid secretion of LH leading to its maximum level during the mid-cycle called LH surge induces rupture of Graafian follicle and thereby the release of ovum ovulation. The ovulation ovulatory phase is followed by the luteal phase during which the remaining parts of the Graafian follicle transforms as the corpus luteum. The corpus luteum secretes large amounts of progesterone which is essential for maintenance of the endometrium. Such an endometrium is necessary for implantation of the fertilized ovum or the events of pregnancy. During pregnancy, all events of the menstrual cycle stop and there is no menstruation. In the absence of fertilization, the corpus luteum degenerates. This causes disintegration of the endometrium leading to menstruation, marking a new cycle. In human beings, menstrual cycle ceases around 50 years of age, that is termed as a menopause. Cyclic menstruation is an indicator of a normal reproductive phase and extends between menarche and menopause. There is a table given here, menstrual hygiene. Menstrual hygiene, maintenance of hygiene and sanitation during menstruation is very important. Take bath and clean yourself regularly. Use sanitary napkins or clean homemade pads. Change sanitary napkins or homemade pads after every 4 to 5 hours as per the requirement. Dispose of the used sanitary napkins properly, wrapping it with used paper. Do not throw the used napkins in the drain pipe or toilet in the open area. After handling the napkin, wash hands with a soap. Fertilization and implantation. During copulation, coitus, semen is released by the penis into the vagina. Insemination, the motile sperms rapidly pass through the cervix, enter into the uterus, and finally reach the ampullary region of the fallopian tube. The ovum released by the ovary is also transported to the ampullary region where fertilization takes place. For fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary region. This is the reason why not all copulation lead to fertilization and pregnancy. The process of fusion of a sperm with an ovum is called a fertilization. During fertilization, a sperm comes in contact with the zona pellucida layer of the ovum and induces changes in the membrane that block the entry of additional sperm. Thus, it ensures that only one sperm can fertilize an ovum. The secretion of the acrosome helps the sperm into the cytoplasm of the ovum through the zona pellucida and the plasma membrane. This induces the completion of the meiotic division of the secondary oocyte. The second meiotic division is also unequal and results in the formation of a second polar body and haploid ovum oocyte. Soon the haploid nucleus of the sperms and that of the ovum fuse together to form a diploid zygote. How many chromosomes will be there in zygote? One has to remember that the sex of the baby has been decided at this stage itself. Let us see how. As you know, the chromosome pattern in the human female is double X or XX, and that is the male XY. Therefore, all the haploid gametes overproduced by the females have the sex chromosome X, whereas in the male gametes, if sperms, the sex chromosome could be either X or Y. Hence, 50% of sperms carry the X chromosome, while the other 50% carry Y. After fusion of the male and female gamete, the zygote will carry either XX or XY depending on whether the sperm carrying X or Y fertilize the ovum. The zygote carrying XX will develop into a female baby and XY will form a male. You will learn more about the chromosome patterns in next chapter. That is why scientifically it is correct to say that the sex of the baby is determined by the father and not by the mother. The mitotic division starts as the zygote moves through the isthmus of the oviduct called cleavage toward the uterus and forms 2, 4, 8 or 16 daughter cells called blastomeres. The embryo with 8 to 16 blastomeres is called a morula. The morula continues to divide and transforms into blastocysts as it moves further into the uterus. The blastomeres in the blastocyst are arranged into an outer layer called trophoblast and an inner group of cells attached to trophoblast called the inner cell mass. The trophoblast layer then gets attached to the endometrium and the inner cell mass get differentiated as the embryo. After attachment to the uterine, cells divide rapidly and covers the blastocyst. As a result, the blastocyst becomes embedded in the endometrium of the uterus. This is called implantation and it leads to pregnancy. Pregnancy and embryonic development. After implantation, finger-like projection appear on the trophoblast called chorionic villi, which are surrounded by the uterine tissue and maternal blood. The chorionic villi and the uterine tissue become interdigitated with each other and jointly form a structural and a functional unit between developing embryo, fetus, and the maternal body called placenta. The placenta facilitates the supply of oxygen and nutrients to the embryo and also removal of carbon dioxide and excretory waste material produced by the embryo. The placenta is connected to the embryo through an umbilical cord which helps in the transport of substances to and from the embryo. Placenta also acts as an endocrine tissues and produces several hormones like human chorionic gonadotropin (HCG) and human placental lactogen, that is HPL, estrogen, progesterone and etc. सॉरी यहाँ पे प्रोजेस्ट्रॉन नहीं है, प्रोजेस्टोजेंस लिखा हुआ है. In the later phase of pregnancy, a hormone called relaxin is also secreted by the ovary. Let us remember that HCG, HPL, and relaxin are produced in women only during pregnancy. In addition, during pregnancy, the levels of other hormones like estrogens, progestogens, cortisol, prolactin, thyroxine, etc. are increased several folds in the maternal blood. Increased productions of these hormones is essential for supporting the fetal growth, metabolic changes in the mother, and maintenance of pregnancy immediately after implantation. The inner cell mass embryo differentiates into an outer layer called ectoderm and the inner layer called the endoderm, and a mesoderm soon appears between the ectoderm and the endoderm. These three layers give rise to all tissues organs in adults. It needs to be mentioned here that the inner cell mass contains certain cells called stem cells which have the potency to give rise to all the tissues and organs. What are the major features of embryonic development at various months of pregnancies? The human pregnancy lasts 9 months due to, do you know for how many months pregnancy lasts in dogs, elephants, cats? Find out in human beings. Find out in human beings after 1 month of pregnancy, the embryo heart is formed. The first sign of growing fetus may be noticed by listening to the heart sound carefully through the stethoscope. By the end of the second month of pregnancy, the fetus develops limbs and digits. By the end of 12 weeks, first trimester, most of the major organ systems are formed. For example, the limbs and the external genitalial organs are well developed. The first movements of the fetus and appearance of hair on the head are usually observed during the fifth month. By the end of the above 24 weeks, end of second trimester, the body is covered with a fine hair. Eyelids separate and eyelashes are formed. By the end of 9 months of pregnancy, the fetus is fully developed and is ready for delivery. Parturitions and lactation. The average duration of human pregnancy is about 9 months, which is called the gestation period. Vigorous contraction of the uterus and the end of a pregnancy causes expulsions or delivery of the fetus. This process of delivery of the fetus, childbirth, is called parturition. Parturition is induced by a complex neuroendocrine mechanism. The signals for parturition originate from the fully developed fetus and the placenta, which induces mild uterine contraction called fetal ejection reflex. This triggers release of oxytocins from the maternal pituitary. Oxytocin acts on the uterine muscles and causes a stronger uterine contraction, which in turn stimulates further secretion of oxytocin. The stimulatory reflex between the uterine contraction and oxytocin secretion continues, resulting in stronger and stronger contraction. This leads to expulsions of the baby out of the uterus through the birth canal. Parturition. Soon after the infant is delivered, the placenta is also expelled out of the uterus. What do you think the doctors inject to induce delivery? The mammary glands of the female undergo differentiation during pregnancy and starts producing milk toward the end of pregnancy by the process called lactation. This helps the mother in feeding the newborn. The milk produced during the initial few days of lactation is called colostrum, which contains several antibodies absolutely essential to develop resistance for the newborn babies. Breastfeeding during the initial period of infant growth is recommended by doctors for bringing up a healthy body. Thank you so much for completing this human reproduction chapter. This is the summary. Thank you. Till then, bye bye. All the best for your preparations.